Hi everybody. In this series of videos we want to talk about the pharmacokinetics and pharmacodynamics of the drugs which are administered within the body. Well, when we administer the drugs in the body, we actually are looking for an optimum window in which the drug has its maximum efficiency but its minimum toxicity. As you see here, when we draw the concentration of the administered drug with respect to the time, you see here that this limit just defines the maximum concentration of drug that can be used to avoid the severe toxicity in the body. However, this limit tells us that if the concentration of drug is less than that, it's not going to be effective and useful. So we have to, let's say, maximize the efficiency of the drug and also minimize the toxicity. Well, now let's talk about the pharmacokinetics. Well, when we administer the drugs, in the case of pharmacokinetics, we just talk about how this drug is going to be distributed and the metabolism and the excretion of the body of the drug in the body is going to be studied. For example, we administer the drug and we want to realize how it is going to be absorbed by different tissues and different target sites, how it is distributed in the body, how the metabolism takes place, and how much of this drug is going to be excreted from the body. So all of these are studied when we deal with the pharmacokinetics. Actually, we also have to take into account that for different patients we have a viability for pharmacokinetics of the drugs. For example, just consider a group of males or females. Because they have different because they have different genetics, so the pharmacokinetics is going to be different. For example, if we assess the urine that we get from the patients, we can assess the different amount of drugs that have been excreted from the body when we have applied a certain amount of drug. Well, we talked about the pharmacokinetics and the concentration of the drug within the body. Actually, the amount of drug that we inject into the body, the amount of drug which enters the blood is different from the amount of drug which reaches the action site. For example, just imagine that you are going to treat the cancer cells. And imagine that you inject a certain dose of drug into the blood, but how much of this drug is going to reach the cancerous tumor? This is something that we want to measure, but practically the direct measurement of the drug concentration at the target site is not going to be that easy. So it is something far from the, let's say, reality. But if we are lucky, we can just say that we administer a certain concentration of drug within the blood, and we want to realize how much of that drug is going to be accumulated in the tissue that we have in our mind. Okay, so we want to find a sort of a correlation between the amount of blood that the amount of drug that we have administered and the amount of drug that is accumulated at the target site. And if we are lucky, this relation is going to be linear. So you see here that this is the amount of drug which is administered within the blood, and here we see the amount of drug within the tissue. So you see here that, for example, the concentration of drug that we inject in our body in the plasma or in the blood is shown here, and it has a relationship between with the concentration of drug which will be accumulated in the tissues. So if this relation is linear, we can define the constant, the slope of this relation. For different tissues, we can see how much of the drug is going to be concentrated in the plasma, for example in the blood, or how much of that is going to be accumulated in the receptor, or how much is going to be excreted out of the body with respect to the time. And you see here in this figure the concentration of drug with respect to the time. Well, besides the pharmacokinetics, we have the pharmacodynamics. In pharmacodynamics, we want to see the relationship between the drug concentration and its efficiency, the amount of drug that is going to be effective for us, how effective the drug is going to be. Well, you know, this pharmacodynamics is going to be much more complex because we have the biochemical cascade process in which if you have the drug, for example, in the affinity of these receptors, first of all, they have to send a signal to the nucleus so that the receptors on the surface of the cell have to be modified. They have to increase in their numbers so that they uptake the drug, and then the drug just do the endocytosis, let's say, and then they are going to be effective. So this is not something easy to be measured. And we just told you that the viability of different patients from patient to patient makes the measurements to be different for different patients. On the other hand, if we just plot the amount of drug in the vicinity of the receptor, okay, which comes from the pharmacokinetics, I told you that from the pharmacokinetics we will get the information about how the drug is going to be distributed. Okay, so just imagine in the vicinity of this cell, if we plot the drug that is in this area in the vicinity of the cell, and the effectiveness of the, let's say, of the drug, we can get some diagrams that you see here. So actually here we have the concentration of drug in the plasma in the blood, and we have the corresponding effectiveness to that concentration. So you see here, if we plot this diagram in a semi-logarithm scale, there is a concentration of drug which is called the EC50. Okay, let me just, the EC50. The EC50 tells us that for the concentration below that we are not going to be effective, but for the concentration above that the effectiveness increases to the point that we reach the 100% effectiveness, and any increase in the drug concentration is gonna lead to the side effects. So the drug will show toxic effects at that point, and this is not something that we like. So you see here that this yellow window shows us the effective therapeutic window. Well, the amount of concentration that you see here at which the effectiveness is not going to be increased by increasing the concentration of the drug is called the Emax. Well, now let's have a look at another now let's have a look at another issues about the concentration of the drug which is administered into the patient. Well, now we want to talk about the tolerance. Tolerance simply says that if I administer the drug into a patient, it's going to be effective at this concentration. However, for the next time, I have to apply more amount of drug, higher amounts of drug to reach the same effectiveness. You can compare it with the people who are addicted to drugs. For example, they start with the cigarettes, then they will improve their drugs. Much more effective and stronger drugs have to be used in order to give them the satisfaction in order to be effective. So this is called as the effectiveness. It also affects the pharmacodynamics of the drugs, because we just mentioned here that the pharmacodynamics deals with the concentration of the drug that has been administered in the body, and also the effectiveness of that amount of drug. So you see here, if we are administering the same drug for many times, the concentration of drug to reach that effectiveness has to be increased. So it is highly affected by these tolerances. Well, as we mentioned that we have the curve about the effectiveness and the concentration. So you see here that we reach the effectiveness that we wanted. On the other hand, you see here that this curve shows how toxic the drug is going to be at that concentration. This is the therapeutic window that we were talking about, because we want a sort of concentration at which not only I'm sorry, because I paused the video for a minute. There was a friend of mine coming, and I cannot remember what I was exactly saying. Maybe I was talking about the therapeutic window. Well, yes, this is the concentration at which we have the maximum efficiency and somehow the minimum toxicity. So you see here that at this concentration, although the efficiency is maximum, but the toxicity is also high. But however, here, although the toxicity is the lowest possible, but the efficiency is not going to be that much appropriate for us. So you can say that this is the optimum window that we wanted to define. So let's have a look at another thing. Well, we mentioned that from patient to patient the viability is different. For example, consider the time that a certain amount of drug is fixed, but for different people. You see here, a different population of people, well, the highest plasma drug concentration is going to be different for different patients. So this tells us that the pharmacodynamics and pharmacokinetics is also highly affected by different individuals. Okay, because there are variations in the drug absorption, there are variations in the drug distributions, and also differences in an individual's ability to metabolize and eliminate their drug. Well, on the other hand, in many cases, the patients have different diseases when this diagram has been plotted. For example, some of them were suffering from renal or hepatic insufficiency, and some of them, other different diseases. But anyway, let's see when the diagrams that we get from the pharmacokinetics and pharmacodynamics are gonna be valuable, are gonna be useful for us. Well, in the first place, there should be a good correlation between the amount of drug concentration and the response and the effectiveness that we get. On the other hand, there should be a wide therapeutic window, the one that I showed you just here. This therapeutic window has to be as wide as possible. On the other hand, the amount of information that we get should come from thousands of patients. I mean, we just cannot say that we have one patient and this diagram coming from one patient is what we can trust. For example, have a look at this diagram, and you see here that the concentration of the drug that has been used with respect and the corresponding effectiveness for different patients finally seems to have a, let's say, linear relationship. But sometimes the diagrams that we get from the pharmacodynamics or pharmacokinetics are not going to be valuable. For example, consider the time that there is not a well-defined relationship between the concentration and the effectiveness. Sometimes some metabolites are produced which can interfere with the drugs, and the value of our models, how our models are going to be precise, can be affected by these metabolites. Sometimes there are some toxic effects which show high toxicity even at low concentrations, so there is no chance to increase the concentration of the drug that we are going to use. So practically there will be no chance for having the clinical trials. Well, and sometimes there are no significant consequences associated with too high or too low levels, so nothing is shown, no efficiency, and nothing happens. Well, in this scheme you can see how the first steps of making the pharmacokinetics diagram starts. You see here that we will have a patient and we will diagnose this, and a drug is going to be selected, and then we have to guess an optimum drug concentration that have to be applied for this patient. And after this drug is applied, by measuring the results, the effectiveness, and comparing with the optimum drug concentration that we have guessed at the beginning, we can realize whether that amount of drug has been useful, effective or not, and then we can finally draw the pharmacokinetics and pharmacodynamics diagrams. Well, we told you that we from the pharmacokinetics, by measuring the distribution, the absorption and the excretion of the drug, we can get the pharmacokinetics, and from that by measuring at the site of action, I showed you that we had the cell, and in the vicinity of the cell we have to measure the drug concentration, and from that, by comparing the effectiveness of the amount of drug which is in this vicinity, we can go to the pharmacodynamics. One of the factors which affect the pharmacokinetics is the compliance. Just imagine that some patients are really not compliant to a certain type of drug. For example, the doctor prescribes 2 tablets for a patient. He takes it, but it's not going to be useful. So this compliance should not gonna affect our model because simply a patient doesn't show any let's say response to that type of drug that has been prescribed. Well, in this table you see the therapeutic range for different drugs. I mean, the range at which they start to have the minimum efficacy until the maximum efficacy before they show the toxic effects. Well, sometimes, in order to understand better how the absorption, distribution, metabolism, and the elimination or excretion of the drug happens within the body, we have to do some simplifications to predict a drug's behavior in our body. To do these simplifications and come up with the prediction, we have to consider some compartments. A compartment is simply, just imagine that it tells us a certain amount of drug just enters, some of them is going to be accumulated here, and some will come out, will be eliminated. So by writing let's say a mass balance for this compartment, we can realize how much the drug has been administered and how much of that is going to be finally eliminated. Well, the amount of blood that is going to be finally eliminated depends on the let's say elimination constants K that I will go for. Okay, now let's talk about the model that we were talking about. We said that we have to consider some compartments. You see here that depending on the vascularization of different compartments, we can group them together. For example, for the blood, kidney, lungs, liver, and heart, because the amount of blood which is distributing within these compartments is somehow the same, we group them together. On the contrary, we have the fat tissues. They have no vascularization, they are less vascularized, so we can say that less amount of blood is going to be flowing there, so less amount of drug is going to finally reach there. So the concentration of blood accumulating, reaching these parts is going to be much lower. But anyway, although considering more compartments is going to be more useful, but it makes our model to become more complex. Well, here you see that the mass balance, for example, for this compartment, tells us that a certain amount of drug just enters in, some is accumulated here and some will come out. The elimination rate constant tells us how much of the blood is going to be let's say eliminated, excreted out of the body, and the less it is, the more of drug is going to be accumulated within the body. Well, and this scheme is just a simplified version of what we saw above this, that we just consider a central and peripheral compartments at which a certain amount of blood enters and some will be eliminated. Well, just imagine, for the time that we just consider one compartment, there is only this. So we administer a certain amount of blood and we can just assume that the distribution of blood within all these compartments is the same. Okay, so once the drug is injected, all the drug is present all over the body at the same concentration. However, if we use two compartment model like this, we can assume that we administer the drug, and after that the concentration will increase in the high vascularized compartments such as the kidney, the lungs, the heart, that we grouped them together as you saw here. Okay, so first the concentration of the drug within these organs is going to be increased, and by the passage of time the whole amount of drug is going to be distributed the same all over the body. I can say that the drug will reach an equilibrium. Well, you can write some differential equations for your mathematical model, the amount of drug entering and coming out, and this K can be identified experimentally, because if you want to identify how much of your drug is coming out, you can use for example, you can assess the urine to see what has happened to the drug that you have administered and comes out. And I think that we are only limited to the urine. There's nothing else that we can do. Okay, let's talk a little about the volume of distribution. Well, the amount of drug that we have applied, we know that a certain concentration of the drug is going to be in different organs. The more the volume of that organ tells us that the less concentration of the drug is. Okay, so by this volume of this distribution, we can realize how well the drug has been distributed. For example, if we consider the volume of distribution to be very high, we can say that the concentration of drug in the blood is low and the drug has reached other tissues. Okay, but if the volume of distribution is less, is small, it means that the volume in which the blood has been distributed is only the plasma. So the volume of distribution is small, so it says that the drug has not been distributed well. So for the small volume of distribution, we have we can say that most of the drug that we have administered has remained in the blood, so we have failed to diffuse the drug in different tissues. Okay, for example, in this table you can see that for different drugs we have different volume of distribution, which tells us how well they are going to be distributed. As you see here, for example, for the amlodipine, I hope that I have pronounced this name correctly, the volume of distribution is much higher compared to the valsartan drug, which is going to be distributed in a lower amount of volume in the body. So most of this drug is going to be remained within the blood and not diffusing into the other organs. And in this picture I just told you about the elimination. We can experimentally measure the amount of K when we administer a certain amount of drug. But now let's talk about the distribution of the nanoparticles. You know that the nanoparticles are encapsulating the drugs. So the model here is going to be a little bit more complex, because not only we have to consider the distribution of the nanoparticles, we have to also consider the distribution of the drug, how the drug is going to be diffused. So there are two factors, and considering these two factors together is going to be so complex. But for the moment, let's just consider the time that we have only the nanoparticles in the system, and let's see how their distribution is gonna be affected. Well, in this table you see different particles that we have, and depending on the different physicochemical properties, for example the surface charge, the zeta potential, the size of the particles, the coating, and etc. So depending on the physicochemical properties, their biodistribution is going to be affected. You see here we have the different organs such as blood, liver, spleen, lung, brain, and etc. And these dots simply tells us if the nanoparticles have been obtained within these organs or not. These dots tell nothing about the concentration of the nanoparticles within these organs. Well, one of the models that we can consider is the blood flow limited model for the pharmacokinetics, and it tells us that within this blood flow limited, it is only limited to the blood flow, and the diffusion of the drug into the tissue is not gonna be hindered, is not gonna be hampered. So the pharmacokinetic tells us that the more the blood flow reaches this site, the more the drugs have the possibility to diffuse into the tissues. Okay, let's talk a little bit about this diagram. You see here that it tells us the charge of the particle from the negative charge is going to be increased to the positive charge. The size here increases from the 1 nanometer to 220 nanometer, and you see here we have the immune system of the body, which simply tells us that if we go on the diagonal here, that not only we increase the size of the nanoparticles, but also we charge them positively, more of the nanoparticles are going to be excreted out of the body. They are going to be recognized by the mononuclear phagocyte system of the body. The reason is that the larger particles, if you remember, are gonna be excreted readily
fast, rapidly out of the body. On the other hand, when the particles are charged positively, the amount of proteins which are going to be observed on the surface of the nanoparticles is going to be increased. So they will undergo the, let's say, I'm sorry, when the particles are negatively charged, they undergo the opsonization. On the other hand, the reason why the positive charge of the nanoparticles leads to higher recognition by the mononuclear phagocyte system is that they will have a stronger interaction with the cell membranes, so they will be recognized faster. On the other hand, even for the negatively charged particles, we have the opsonization. So the opsonization just excites the phagocyte cells for excreting the particles out of the body. Well, here in this scheme you see that if we equip the nanoparticles that we have with a certain type of fluorophore that can be recognized, identified under the fluorescence microscopy, if we inject them into this mice which has a tumor on its surface, we can study the pharmacokinetics and pharmacodynamics of the nanoparticles inside this tumor. But this slide is more important for us. Well, we want to just say that when the particles enter the body, they are faced with a sequential biological barriers that they have to overcome to increase the efficiency of the drug. Because we said that the pharmacodynamics and because we said that the pharmacokinetics is gonna be increased the more the nanoparticles or the drug can be present in the vicinity of the target site. On the other hand, if the presence of the drugs in the vicinity of the target site is going to be increased, then the pharmacodynamics has the chance to be increased as well. So, for the particles, in order to reach higher pharmacokinetics and higher pharmacodynamics, they have to overcome a sequential biological barriers which are trying to get rid of the nanoparticles within the body. Well, the first thing is the opsonization that happens. When we when we inject the nanoparticles, the proteins are going to be let's say observed on the surface of the nanoparticles, and their recognition by the mononuclear phagocyte system of the body, and they will be eliminated out of the body. On the other hand, some studies show that I'm gonna talk about this effect in this slide. Let's have a look at this here. Some studies show that the shape of the nanoparticles also affect their biodistribution, because the fluid dynamics shows us that we have the focusing region here, that the particles tend to accumulate in the central part of the fluid flow, and less of the particles like to be in the margin. Okay, so we can increase the margination effect. Let me see if I can find it. Yes, it's here. the margination effect by tuning the shape of the nanoparticles. What do we mean? If we change the shape of the nanoparticles from the spherical one to the disc-like one, they will tend to marginate in the vicinity of the endothelial cells. So now they have more chance to internalize. On the other hand, there are some receptors on the cell surface. If the particles are in the disc form, they will have more surface to connect to these receptors, so the adhesion is gonna be increased. All I said, you can find them here in this paragraph. Well, then let's talk about the EPR effect, because EPR effect also affects the biodistribution, and we know that let's say enhanced permeability and retention effect helps us to increase the amount of nanoparticles which are going to be finally accumulated within the body, within the cancer tumor. You see here that for the mice and animal models, because we induce the growth of the tumor very fast, they are less vascularized, and there you can see more fenestrations here, so there is more possibility to enter the cancerous tumor. But for the normal cancerous tumors, it's not going to happen. I mean that the vascularization is more compact, is more, let's say, ordered. You see less fenestrations, and it decreases the EPR effect. To overcome this problem, we can accommodate our particles with these metalloproteinase MMP2 and MMP9, which will degrade the extracellular matrix of the cell membrane and make it more permeable. Well, let's jump to the last slide that we are going to talk about. Okay, before that, let me just tell you that also the blood-brain barrier is another barrier that the nanoparticles have to overcome. And if you remember, we had a series of videos about the blood-brain barrier, which told us that to ensure the maximum security for the blood, you see different layers of the endothelial cells which do not let the nanoparticles to enter here and pump them back into the bloodstream. Okay, let's talk about the last slide here. We want to say that one of the ways to decrease the opsonization effect on the surface of nanoparticles was using the PEG to pegylate them. There are three more strategies that we never talked about. One of them is to, let's say, cover the surface of the particles with these CD47. Let's say I want to see, I don't know exactly what this is. Let me just, peptides. I was forgetting the name. The CD47 peptides. These CD47 peptides are the receptors on the surface of the cells. If we accommodate them on the surface of the nanoparticles, when they come close to the phagocyte cells, they see the do not eat me signals. so they will not be eliminated out of the body. Another strategy is to coat the surface of the particles with the cell membrane of the macrophages, as you see here. So when we use the cell membrane of the macrophages will consider these nanoparticles as being self, as being someone like them, not a different type of person. So again, the elimination will not happen. And finally, the red blood cells, if we use the cell membrane of the red blood cells, again the nanoparticles are going to become more biocompatible in the body. And also the coatings are biodegradable. But although these three strategies are going to improve the coating and the biocompatibility of the particles, the problem is with the techniques that we have to use to stick, to attach, to conjugate the membrane on the surface of the particles, which is going to be much more difficult compared to the polymeric PEG chains that we can use. Okay, this was all about the there is one more thing here, the Elvis. Let's talk about the Elvis, and then we will finish this video. Well, Elvis is the extravasation through leaky vasculature and inflammatory cell-mediated sequestration. It simply tells us that when a tissue is inflamed, different, let's say, growth factors, histamine will be released, and because of that the tissue is going to be more permeable, okay, and the release of these factors make the tissue to become permeabilized. Well, when this happens, the nanoparticles then can enter the tissue. You have to consider that the inflamed tissues, there are too many macrophages. I can show you here. The macrophages are the phagocyte cells, and when the particles enter there, they will be uptake by the macrophages and they can accumulate there. Then after that, the drug can be released or diffused out of the nanoparticles and treat that inflamed tissue. So all we wanted to say about all of these examples, you saw the Elvis, the APR effect, the margination, the coating, and these sequential barriers, just they were intending to give you one message, that the pharmacokinetics and pharmacodynamics of the nanoparticles and the drugs which is encapsulated on their surface or within them is highly affected by different factors that we talked about. The charge, the physical chemical properties, the barriers, the coating, the margination, the shape, the APR effect, the Elvis, and everything. So this was all about the introduction to the pharmacokinetics and pharmacodynamics, and I hope that it has been useful for you. Okay, you can download the notes so that you don't have to take any notes. I have written all you have to know in the notes. Okay.